All right, welcome back. Now let's talk exceptions. Or maybe just things that are at the fringe of what is possible and kind of show us what you can do within the constraints of a virus like lifestyle. Yeah, that's about as definite as I can be about any of this. Let's go. So, what if you had a virus and it just didn't make a capsid protein anymore? The capsid protein is there because it enables us to make a nice little virus particle like this, virion, and that's the thing that's going to spread from cell to cell. But if you had some other way of spreading cell to cell, like in fungus, they have these hyphae, which are these sort of single-celled networks of tubes that are going to connect up multiple cells and potentially share cytoplasm and all kinds of crazy stuff. Theoretically, you could just get into that once, and then even if you lost your capsid, the virus could still spread around. Interestingly, we see this with some viruses. With coronaviruses, for example, once they get into a mouse, they can stay in that mouse for as long as the mouse is alive, something like a year for the regular kind of lab mice. And during that time, the virus will keep on replicating usually, but it will often lose some of the genes that it needed in order to get in the first place, because now it's replicating in a place and in a way that actually doesn't require it to spread from cell to cell like it used to. Very interesting, I think. So hypoviruses are viruses of fungi, and they're going to spread through hyphae, as far as we know. They appear to be spread as just naked RNA, and they do encode their own polymerase. And that's this big gray gene that's up here, and a couple of maybe cofactors for the polymerase, a couple of proteases by the look of it, the two blue ones. But they don't encode a capsid per se. So they're going to spread kind of the way that viroids would spread, just as naked nucleic acid. And you wouldn't think that would be a good way to do it because they're RNA, and RNA is destroyed pretty readily in this world. But double-stranded RNA is pretty stable, and so as long as they, sort of fold up like double-stranded RNA, then presumably they can go wherever they want. Yeah. Shown down here at the bottom, these are circoviruses. There are a lot of circoviruses in the world. They can kill you through diarrhea. That's one of the famous things that they can do. So these are very little viruses. They're single-stranded DNA. I believe Torquatino is one of these things. And these things are they're kind of like the minimal virus, and maybe we should have started with them. But they're going to have a capsid protein, which makes a nice little capsid here, and it's a T=1, which is the simplest type of capsid. So it's just a bunch of little triangles that pack together, and all the triangles make a 5-fold axis. You get 5 of those to make 5-fold. You don't need any VP2 and VP3. It's like if you made a picornavirus with just VP1, just the 5-fold axes. So they make a tiny little virus. And since it's a tiny virus, it can only hold a tiny genome. And here's your tiny genome. It's an RNA-dependent RNA polymerase, and it's a capsid gene. And that's it. It doesn't encode anything else. And so those are somewhere close to what I would consider to be a minimal virus. But let's go on to some weirder viruses. So what about satellite viruses? So in this world, there are several viruses, and we don't know as many of these as we know of viruses, but these are things that are viruses of viruses. So they require not only things from the cytoplasm of a cell, but also they require components from another virus, a helper virus that probably doesn't want to help, but, gets roped into it anyway. Yeah. And so here's like an alpha satellite virus up here. And these things will steal capsids from Gemini virus, for example. Or they can steal capsids from nanoviruses. And they are good enough at slipping themselves into these capsids. It's kind of like some of those birds that'll put their eggs in other birds' nests, and then the baby will sort of kick the other eggs or chicks out of the nest so it can grow really big, really fast, and parasitic. Very parasitic, yeah. But it's a virus that's parasitizing different kinds of other virus. And so this can only work if they are always co-transmitted. And they're going to be co-transmitted if they get into the phloem. That's like where all the sugar transport happens in a plant. It's like the blood of a plant. If blood didn't have to like flow around with a heart and keep you alive, it's actually not like blood at all. But it's nutrient transport. It's the liquidy stuff that's inside of plants. It's like sap, I guess. Yeah, I guess. I don't know. Although now it's not even exactly sap. It's phloem. Yeah, look it up in biology textbooks. And if you get viruses that get into there, then when a biting insect or like a sucking insect is slurping out that delicious phloem, then it's going to pick up a whole bunch of viruses. And when it goes to feed on another leaf, stem, or whatever plant, it's going to dip its dirty needle back in there, and they'll usually regurgitate a little bit of what they just ate, because insects are gross people. Yeah. And so this is how we get arthropod-borne diseases, mosquito-borne viruses, things like that. Exact same mechanism, just in people. And so, yeah, that is how it works. So alpha satellite viruses encode their own RNA-dependent RNA polymerase, or replicase at least. Sorry, it's a DNA genome, so it's a DNA polymerase. But they don't encode anything else. They steal their own capsids. Yeah. So they have to be transmitted. So it puts some limitations on where they can go and how they can be transmitted. But they're doing fine in the world. And as long as there are plants that are tasty and insects that like to taste them, you'll probably get stuff like this that's happening. Yeah. Now for a really crazy one. Now for something completely different. This is nanovirus. Nanovirus, yeah, we met its capsid in the last slide. Little icosahedral, T = 1, teeny tiny little icosahedral thing. It encodes its own protein. It encodes its own polymerase. It's a little single-stranded DNA genome. And it encodes a whole bunch of other proteins. The MP is going to be a movement protein that helps it to get into the vasculature, into the phloem of a plant, which helps it travel around throughout the plant and also helps it to be in the right place for when a biting insect is slurping that delicious good stuff. It can then get transmitted on. These guys, though, have taken the insane step, and yeah, insane, of fragmenting their genome into 8 parts. And whereas the flu virus has 8 parts that evolved a way to hold them really closely together, with all these little RNA loops and proteins interacting with RNA and with the other proteins. So flu viruses are very good at counting to 8 segments every single time. These guys appear to just package 1 segment at random per capsid, flood them out into the world, and the thing is, if you don't get all the segments delivered from this plant to the other plant by the biting insect, then you probably have no virus. And yet, this happens enough that these things exist in the world. So as far as we know right now, these are only viruses of vascular plants, and they are going to be vectored. They're going to be carried along by insects, biting, sucking, whatever, insects. Yeah. Viruses. It's a living. Yeah. But a weird one. Yeah. So, no, absolutely insane way to do anything. Okay. Viruses. These don't look like viruses, and they're not. These are cyanobacteria, which don't you dare let me hear you calling blue-green algae, because algae are eukaryotic. They aren't blue and they aren't green, and they're bacteria. So cyanobacteria, yeah, get it right. Come on, you're better than that. Prochlorococcus and Synechococcus are shown here. These are 2 of them. These are cyanobacteria that happen to be infected by a bacteriophage that does a cool thing. The next slide is going to show you the origin of every zombie movie ever, and the slide after that as well. So let's get into it. All right. These are some of the viruses that infect those cyanobacteria. So they have their own cyanobacteriophage, and they look like little tiny lunar landers with short or long little tunnels coming out here. Fine. Yeah. Those are bacteriophage. This is the world. They're not in the Myoviridae anymore. They're probably in 4 different families. And that's fine. The new world is better than the old. Interestingly, these things encode a lot of genes that you don't normally find in bacteriophage. So, You'll find things like, let's see, things for phosphate uptake, which the virus, the bacteriophage, kind of needs for there to be phosphate, but it doesn't normally help the cell to take up phosphate. That's a weird thing. Viruses generally don't help. They just raid and move on. Yeah, because honestly, yeah, who's going to stop them? You? Yeah, I don't know. Yeah. But they also encode several parts of the photosystem that are needed for photosynthesis. So this PS2 is part of photosystem 2. They've got genes that are involved in synthesizing heme groups, which are also used in some photosynthetic processes, and in just electron transfer in general. Things like cytochromes are going to have heme groups in them. And those are weird. What are they doing with these? Yeah, here's what they're doing with them. So this is an example of photosystem 2. Here's photosystem 1 over here. And so there's all these different proteins, and they're going to basically pass electrons down this sort of chain. And each time the electron is passed, it's going to do a little bit of work. So we're going to make some product that stores energy, or we're going to convert something to a different form. It's really neat stuff. It's really bio 2, maybe, stuff. And you know what? That's fine. This is not that class. You don't have to remember that for here. What you got to remember is that this virus makes a thing called a PSBA gene that encodes a D1 protein, and D1 is right here at the catalytic center of photosystem 2. This is the electron capture thing that is holding on to the chlorophyll at the center where all the other pigments get, or where all the electrons get transferred. This is a thing which in no way helps the bacteriophage, and definitely this is host biology specific, and yet the virus is carrying it. Also, there's a thing called the HLI, highlight inducible protein, and these are chlorophyll carriers, and they're essential for growing under a high amount of light conditions. What you see, though, is that when the cells are infected, the cells start to die, as do we all. Yeah, memento mori there for your Monday or whatever day of the week this is. And so as the cells are dying, they're switching off some of their gene production. And so this is the cell's own version of the PSBA, or rather the D1 protein. And see, it goes down, 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 and it gets to about half the normal amount. This is the viral version of that. And as the host version goes down, darn it if the viral version doesn't go up, 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 actually to levels that are surpassing those of what the host was making. So as the host is falling apart, as its genes are ceasing to keep it alive, the virus is putting it on life support. And why would a virus put cells on life support? And it's doing that with the HLI protein as well, which these bacteria encode. It's because the longer the virus can keep that cell alive, the more energy comes into the cell, which is converted into nucleotides, which is going to be converted into brand new copies of viruses. And so these particular viruses that zombify the cells actually end up squeezing a few extra rounds of replication out of the cell before the cell dies. Yeah. Very forcible light support. Here's another one, the D1 protein being replaced by the phage-encoded version of that. So that's pretty neat. And if zombies were real, I think they would maybe work something like this. You'd have to have some sort of thing replacing and subverting the intent of the original biological systems, while also kind of breaking enough of the sort of command and control to make the thing chaotic. Yeah. Without making it just fall apart and die. Yeah. Yet. Yeah. Until it infects more. So, hey, cool, right? Yeah. Zombies. Yeah. Viruses? I don't know. There we go. All right. Now, one thing viruses don't do is encode ribosomes, but viruses do encode parts of ribosomes, and quite possibly, we don't have an awesome paper showing why and how it happens like in that previous paper, because that's really neat. What we have are a bunch of these things, and the virus versions are up here at the top. They're related to each other. There we go. And here are a bunch of other cellular versions, and yeah, they all match up. This is a virus that's stolen a protein from its host, and it's giving it back. Is this, yeah, these like terrible landlords or something? They steal something that belongs to the host, and then they give it back as a gesture of goodwill. Really cool, yeah. Viruses, yeah, they're neat little guys, yeah. Messed up ethics, but yeah, they do some neat stuff. Okay. What else? What else shouldn't viruses do that they totally do? Viruses have no real reason to encode tRNA synthases or tRNA genes for that matter. And yet, in some of these viruses, you will find tRNA gene clusters. Now, these are the clusters. This is the codon that this tRNA gene is going to make. But these are things that can be translated that make a transfer RNA gene. Not translated, they can be copied, rather. Yeah. Look at me messing up my biology 1 terms. And so, yeah, it's a gene cluster. These are neat viruses. These Phycodinaviridae actually have something like, they have a membrane inside. All their DNA is inside of that membrane. So that's kind of like a nucleus. It's a bunch of proteins and double-stranded DNA of the virus in there. And then instead of having like a cell wall like a plant or a fungus, they have a capsid on the outside. And that seems like it's serving the same sort of function as a cell wall. Kind of interesting. Yeah, these things will encode very often some of their own tRNAs. And they may even encode the tRNA synthase is the protein that will add that amino acid onto the tRNA. So it's like charged up so it can do its job in translation. Not things that viruses would normally carry, and probably, even though it looks like they're being nice to the cell, probably this is part of some kind of backhanded, just under the table activity that's going to result in the cell producing more virus in the long run. Because those are the viruses that survive the best, are the ones with the best survival skills. That is a very circular bit of reasoning, and it's also, pretty correct. Now, if you're talking viruses that mess with the cell cycle, it's probably easier to say they all do, but they all do doesn't get you any points on the exam. So here are some very specific examples, and you're welcome to have a look at this. Some of these things we have covered before. So PRB and the cyclins and the cyclin-dependent kinases, those are things that are going to be manipulated by things like human papillomavirus, but also there are other genes from other viruses. So adenoviruses make an E1A protein, and those are going to be involved. They're interfering with P21, which you remember is going to be involved in one of these checks that stops the cell cycle from progressing from one stage to another, at least in creatures like us, etc. So these are places that you can look if you want to build up a really nice story about how viruses just mess with cells in a way that makes more viruses and hurt cells. But yeah, really, do eukaryotes have feelings? I don't know. Yeah, we're a very virus focused class, and so that's okay. We can keep our virus focus. And that may be, that's the end. All right. That's the end of this particular lecture. The subject goes on and on and on forever. I'm just going to stop talking about it for now, and you can pick up where I left off. There's a lot of work to be done here. So thanks very much. Hope you got something weird out of this. That's the best you can ever hope for with viruses. You get something weird. All right, bye.